Hello, I'm Richard Mayeux, and I have the privilege of leading you through a global view of Alzheimer's disease genetics. My colleagues and I will try to give you an informed symposium on the importance of genetics discovery for Alzheimer's disease throughout the world. As we all know, the prevalence of Alzheimer's disease is increasing worldwide. Every continent and every country has experienced an increase in the number of individuals with this diagnosis because our population at large is also increasing in age. The first leg of our tour will start in Europe, where Jean-Charles Lambert will discuss the frequency differences across Europe and also point out to us that the risk, even though the allele frequencies are different, seem to be relatively homogeneous across several European countries. Sudha Seshadri will then describe efforts to investigate the genetic variants across India, China, and other Asian countries. This is important because these populations have been relatively neglected in the past, and our hope is to identify the variants in these populations now to contrast them with what we know from the white non-Hispanic populations in Europe and in the United States. Hugh Hendry will also look at populations that are important to investigate: African Americans, both in North and South America, and the African population. He will describe the early studies, and that will be followed by Peggy Vance, who will share with us the latest results from the largest African American genome-wide array led by Brian Kunkel and Christiane Reitz. This is built off an earlier study by Dr. Reitz and her colleagues, where a smaller study in 2013 identified ABCA7 as an important variant in Alzheimer's disease, important variant in the risk of Alzheimer's disease. Joe Lee will take us to Puerto Rico, and the reason he's there is to study a very unusual group of families that have a mutation in presenilin 1, the G206A mutation, which we believe to be a founder mutation. The interesting thing about this group of families is that the variation in the age of onset can be as much as 20 to 30 years in some families. Joe and his colleagues are now trying to identify what the modifying factors are associated with G206A that might account for this difference in age of onset. Giuseppe Tosto is also investigating the Hispanic population, but taking a different strategy. We know that there are clusters of Hispanics in the Caribbean islands, and there are also populations in Mexico, Colombia, and other parts of the Caribbean, Central, and South American populations. They have an unusual admixture, including Native American, European, African, and Latin American. So these studies are very important in identifying the spectrum of genetic variants in the Hispanic population. Yaquil Quiroz will tell us more about the presenilin mutation in the Colombian cohort, which we've all heard about. She will tell us about the mutation that increases the risk to virtually 100% certainty. We also know that the kindred is large, has a little bit more than 6,000 family members, with a median age of onset of cognitive impairment at age 45 or 44, and the onset of dementia at age 49. They will describe clinical, cognitive, and biomarker studies in this population and how they relate to the onset of sporadic Alzheimer's disease. There are also some works in progress around the world that won't be presented in detail tonight, but I wanted to just give you a flavor of the progress we're making in our worldwide approaches. A new study in Reykjavik, it includes several thousand individuals, and some of whom will receive MRI, CT, and other studies that include deep phenotyping for genome-wide and exome sequencing, as well as a nested case control that will have whole genome sequencing that will be shared with other colleagues worldwide. Similarly, my colleague Margaret Perakek-Vance has begun a study in Peru to investigate that population, which has a bit more native ancestry, and will try to understand the genetic basis of Alzheimer's disease in that population. And so we're looking forward to hearing more about that in the future. So what can we learn from a global approach to Alzheimer's disease genetics? Genetic differences between populations can best be described as the relative frequencies of different alleles at each locus. Decades of research in population genetics have found differences among populations for a large number of genetic loci, including many that are health or disease related. But complex diseases such as Alzheimer's disease are probably the result of converging and diverging processes derived from multiple inputs of both genetic and environmental factors. And I think we all believe that Alzheimer's disease is inevitably the result of interactions between genes, genes with environments, and maybe even environments with other environments. Only by understanding the genetic and phenotypic variability worldwide can we hope to develop a rational treatment or preventive strategy for Alzheimer's disease. I thank you, and I hope you enjoy the conference.